Freshwater fish are a critical source of micronutrients and income for many rural communities across the world. Fish provide nutrients that are highly bioavailable, which means they are easily absorbed by the human body. In the Kenya part of Lake Victoria, rural fishing communities rely on freshwater fisheries for income and food. In these communities, fish are the main and sometimes the only animal source food consumed. Men do most of the fishing, while mainly women are tasked with the processing, drying, and cooking of the fish. Unfortunately, fishing pressure has increased, and the increased competition for fish might be affecting local livelihoods and food security. The University of Stirling in Scotland and the Institute of Marine Research in Norway have partnered to design interdisciplinary methods for understanding how fish catch and consumption in the Kenyan communities around Lake Victoria is affecting people's food security. In this project, we're asking questions like, what happens to fishers' diets when the fisheries start to fail? Are fishing communities transitioning to other livelihoods, and if so, will they change what they eat? Will they eat less fish, perhaps abandoning traditional recipes for convenience food, as we see in other parts of the world? And what does that mean for their micronutrient intake? These questions are particularly interesting in contexts like Lake Victoria, where aquaculture is on the rise. Does cultured fish provide a substitute for wild-caught fish in fishers' diets? Does aquaculture support fishers' transitions to alternative livelihoods? Our partnership was funded by the Marine Alliance for Science and Technology for Scotland. This grant funds an exchange, where researchers are working side by side to develop methods that will contextualize the micronutrient composition of these food samples within the diets of the people consuming them.
I think it is of utmost importance to work interdisciplinary if we are going to reach SDG 2, zero hunger, and 3, good health and well-being. Interdisciplinary work can be challenging, and in my experience, it often takes time to learn each other's languages, jargon, and ways of thinking.
Hi, I'm Beth, a PhD student at the University of Glasgow. Here's a 3-minute talk about my research and exchange visit funded by the Saltire Emerging Researchers Scheme. That's 10 seconds already. Let's get going. Carbon, the building block of life, is constantly on the move around our planet, from the atmosphere to the ocean, to soil and living organisms. The ocean is a major sink of carbon, meaning it takes up more carbon from the atmosphere than it gives out. Atmospheric CO2 dissolves in surface waters of the ocean, where it is biologically fixed into organic carbon by phytoplankton and other macroalgae through photosynthesis. A fraction of this carbon sinks to the sea floor, where it is buried in accumulating sediments. In addition to marine sources, terrestrial organic carbon delivered by rivers or glacial runoff also contributes to carbon storage in marine sediments. If undisturbed from activities such as dredging, carbon can be stored in marine sediments for millions of years. That's cool, but why should we care? Well, marine carbon storage, also known as blue carbon, provides a vital service to our planet by regulating levels of atmospheric carbon dioxide. Blue carbon is therefore a nature-based solution to mitigating climate change and can help us to keep our planet cool. Something that scientists, including myself, are still trying to figure out is how climate change, and in particular, a warmer and more acidic ocean, is affecting carbon storage in marine sediments. To help address this, I'll be visiting Umeå Marine Science Centre in Sweden as part of my Saltire exchange visit. I'll be conducting mesocosm experiments using their state-of-the-art facilities on sediment cores collected from the sea floor and investigating carbon burial in marine sediments. I'll test what happens to carbon when it lands on the seafloor under warmer and more acidic ocean conditions. How will rates of carbon storage be affected? Will microbes thrive or be in distress under warmer and more acidic conditions? And how will this affect rates of carbon remineralization? Which microbial species are responsible for carbon breakdown? By addressing these questions, we will be able to determine the future potential capacity of marine sediments to act as efficient carbon sinks and continue their key role in mitigating climate change in a changing world. Thank you.
I'm Emily Haig, a PhD researcher based at Heriot-Watt University and one of the awardees of the MAST Saltire Emerging Researcher Scheme. My research focuses on marine mammals, that's whales, dolphins and seals, and how anthropogenic activity, i.e. humans, affects them. One aspect of my work explores the potential effects of small vessels and craft, like speedboats, paddleboards and fishing vessels, to marine life. I've been working on this in the coastal waters of Scotland, but as part of the Saltire funding, I got the opportunity to expand my horizons and visit a European-scale H2020 project known as the Saturn Project, which is working to better understand underwater radiated noise from large vessels. To do this, I visited two Saturn-associated research institutions, MARI, the Coordinating Institute of Saturn based near Cork in Ireland, and the Marine Bioacoustics Lab within Aarhus University, Denmark. I got the opportunity to connect and meet with some of the researchers and colleagues connected to the Saturn Project, including Saturn Project Coordinator Jerry Sutton, who walked me through the multi-institution project, the goals, the plans, and the progress made so far. Saturn Project Science Communicator Amy Dozier then took me through the project's outreach strategy and showed how she develops a fantastic array of multimedia resources, including illustrations, videos and newsletters, which provide a platform for the cool research being undertaken within the project to be shared and translated into the world of public and policymakers. I also got to meet many fantastic researchers working on the Saturn Project, including postdoctoral researcher Dr. Charlotte Finlay, who's based at Aarhus University, and is modelling how a reduction in vessel speed can reduce the amount of underwater noise produced and therefore might reduce their overall impacts on the marine soundscape. Also based at Aarhus, Dr. Laia Rojano Donate is using physiological data from tags to explore the energetic consequences of vessel disturbance on Europe's smallest cetacean, the harbour porpoise. To encourage networking and collaborations between Scotland and European institutions more widely, I coordinated workshops in both Ireland and Denmark, inviting along researchers from all visiting institutions and further afield, welcoming all career stages. We discussed how our past or present research relates to the topic of human impacts on the marine environment and spent a day getting to know one another's research, sharing ideas, building new connections, refreshing old ones. These workshops connected people across countries, across institutions, despite different study species or topic. We were all united in a collective interest in understanding and protecting the marine environment. Since the workshops, the new connections forged have led to attendees across institutions working together on multiple publications, funding, fellowships applications, sharing data. It just goes to show the power and importance of getting people together and being excited and open about the science that you do.
Hello everyone, my name is Hannah Ewan. I'm a PhD student at the University of St Andrews and my research focuses on carbon transport and Scottish sea loss. Anthropogenic activities have led to an increase in detrimental gases such as CO2 in the atmosphere. Gases such as CO2 cause climate change, which has severe impacts across the entire globe. A range of technologies are being developed to limit the amount of CO2 in the atmosphere. But we also have natural systems that do some of this and need to be fully understood and protected. On land, for example, trees take CO2 from the atmosphere, convert it to organic carbon and use it to grow. They can also transfer the carbon to the soil. In the sea, we have sedimentary carbon sinks. Sediments store large amounts of organic carbon. This carbon comes from many different sources. At the sea surface, phytoplankton take up CO2 just as the trees do on land. All marine organisms are made up of organic carbon. However, when these organisms die off, they sink to the sea floor. The organic carbon is super tasty to bacteria on the seafloor. However, this can result in CO2 being released back into the atmosphere and contributing to climate change. So we need organic carbon that is not as tasty to bacteria, and preferably a lot of it so it becomes buried faster. So another really important source of organic carbon to the sediments comes from the land. When it rains, some of the organic carbon stored in the soils gets washed into rivers, and from then gets transported to the sea and the sediments. Because of where this carbon comes from, and the processes that it has already undergone in the soil and river, this carbon is less tasty to bacteria and can be stored effectively in sediments. Scotland, sea lochs are incredibly important natural sedimentary carbon sinks. My study area, Loch Etive, is surrounded by peatlands and peaty soils which are very rich in organic carbon. On the west coast of Scotland, it rains a lot, and the organic carbon gets transported by rivers into Loch Etive. But what happens afterwards? Let's have a look inside of the loch. The peaty organic carbon is not tasty to bacteria, so we are particularly interested in what happens to it when it enters Loch Etive. We know that a lot of carbon is stored in Loch Etive, but how much of the carbon that enters Loch Etive stays in Loch Etive? How much goes further, and what role exactly does circulation play in where the carbon goes? Models can help us understand such processes. The Saltire Fund enabled me to visit TU Delft in the Netherlands, where I developed a model for Loch Etive using the modeling software Delft 3D. This model shows how water moves throughout the loch and will be used to assess how carbon moves throughout Loch Etive.
Hi, my name is Magnus. I'm about to start my second year as a PhD student based at both Edinburgh Napier University and Harriet Watt. And as part of my project, I went to the west coast of Sweden to assess the biodiversity and ecological status of natural European flat oyster beds using soundscape analysis, eDNA, and visual surveys. This was done in collaboration with the Swedish Environmental Research Institute, IVL. European flat oysters are important as they filter the water, store carbon, increase biodiversity, and more. These flat oysters used to occur in dense beds throughout Europe, but have been declining severely during the last 150 years, mainly due to overfishing. And therefore, restoration is needed. So critically, to date, there is no effective standard method for rapidly evaluating restoration success, and that's where this project comes in. Since Sweden have denser natural beds than Scotland, it was of great value to be able to investigate these. So, in Sweden, I was first identifying beds of different oyster densities, and along the density gradient, I studied the ecosystem visually, acoustically, and by using eDNA, which in short is short for environmental DNA. So visually, we counted the species present along 10-meter transect lines, and later this data will be analyzed by, for instance, looking at species richness, and diversity indices. And by using an underwater hydrophone, I listened to the soundscape around the oyster beds over several weeks, both in spring and summer, and the recorded sound will then be analyzed by looking at the complexity of the sound, as well as sound pressure levels at different frequencies. And thirdly, by sampling eDNA, it can be estimated from shed genetic material what species that may be present in an area. And finally, by comparing the different methods, it can be determined what pros and cons there are with the different tools and how they can be used together to generate an effective rapid assessment tool, which hopefully could be helpful for the oyster restoration projects all over Europe. Thank you very much.